disorders involve sex cells. Cytogenetic disorders involve sex cells, sex chromosomes, Klinefelter, and Turner syndrome. Klinefelter, do you know what is the karyotyping of Saleh, please?
Syndrome because it's like extra X chromosome. That's in this human have 47 chromosomes. This have a lot of drugs that may be we can see this patient like FSH hormone increase, steroid hormone increase, and testosterone increase.
Come down, yes.
We have a phenotype in the body of the patient like its muscles and maybe a small penis and small testes, and we have large border of hip bone. This syndrome is caused by both
Maternal?
or can we can both from father and maybe from the ex.
Yes. Thank you. Any comment?
Speaking or development of the language and also psychological symptoms like depression and also social skills. And also we have physical features like borders of the hip and also measure of the tissue of the breast and reducing muscle mass.
Thank you. Thank you, Saleh. Thank you. Sokaina, yes. Sokaina, yes. Thank you, Sokaina. Thank you. Saleh, any comment? In Klinefelter, they are male with at least one extra X on the sex chromosome, on the karyotyping. Sometimes they have more than one X. This is because of non-disjunction. It's the most common of hypogonadism, infertility in the male. An extra X can come from mother or father, no difference. Sometimes they have mosaic genotype. For example, they have 46 XY, 47 XXY, or more X. When they have 46 normal XY, the symptoms become milder. Here, you can see normal fetus, Y and X. But in this picture, the sperm is XY because of non-disjunction. The ovum is X, so we have XXY, Klinefelter. Here, abnormal ovum with normal sperm. So we have again Klinefelter syndrome here. This is the genotype by karyotyping. Saleh and Sokaina explain about their clinical features. They have some increased autoimmune disorders, mean that my body produces antibody against my body, my cells. For example, they may have systemic lupus erythematosus. Do you know what is system? Yes?
It is related to the antiphospholipid antibody syndrome.
Some of them related to antiphospholipid antibody. Um, in system, do you know what's their phenotype? What's their problem? What's their chief complaint? They have skin. They have skin problems. For example, they have mallow rash. They are photosensitive. Yes, because of autoantibody go against renal. They have glomerulonephritis. Yes, they have serositis. They have serositis, carditis, pericarditis, peritonitis, and inflammation of serosal. Yes, that's right. Increase the clotting in their body, and they have some ulcers inside the mucosa, inside the umbilical mucosa. In puberty, systemic? No, I don't think so. Okay. In Klinefelter, the seminiferous tubules become atrophic and hyalinized. Hyalinized mean hyalinization. Yeah, means something like amorphous eosinophilic material deposit inside the seminiferous tubules. So Leydig cells become prominent. They are prone for germ cells tumor. They have risk of breast cancers. They mildly are mental retardation. They have mild mental retardation, small testicles, bigger breast, hypotone muscles, weaker bones, and Turner syndrome. Who can explain Turner syndrome for me? Turner syndrome? No? Yes. What was your name? Ahmad. Ahmad, huh? Ahmad Abbas. Ahmad Muhammad. Where are you? Muhammad Ab Hussein? Okay, Ahmad.
Turner syndrome is a chromosomal syndrome that affects females that has it by the loss of the autosomic chromosome X that causes 45X karyotype. It caused by loss or complete loss or partial loss of the X chromosome, and they have the normal karyotype self and the other have 45 karyotype chromosome. And it has some feature like lack of the secondary characteristic, sexual characteristic, absence of the menstruation, can have some skeletal issue, have can cause lymphedema, some cardiac issue, and renal issue.
Yes, thank you, Dr. Saleh. Could you please open the windows? Thank you. Okay. Turner syndrome is one of the most common cause of infertility in females. Because of absence of chromosome, one of the chromosome X, XO, in one type, entire X is lost. Sometimes we have mosaic pattern. Some of the cells are genotypically normal, and some of them are abnormal XO. And also we have some cytogenetic abnormalities, such as we have ring chromosome in some terners. We have deletion in P or Q. We have isochromosome of Q, mean that deletion of P arms, isochromosomes. This is mosaic, normal, and missing X chromosome in some cells. In mosaic, they have milder symptoms. They have growth retardation. They are sometimes impotent. They have lymphedema on their limbs, on the neck. When you look at the posterior hairline in Turner, it's down, low posterior hairline. They have horseshoe kidney. You know, what does it mean, horseshoe? From in upper pole, two kidney are connected. They have amenorrhea and hypothyroidism. They have wide-spaced nipples and breasts. They have abnormal type of structure of hand, cubitus valgus. Cubitus valgus. Cubitus valgus. And they have atrophic ovaries. Atrophic ovaries. Karyotyping in Turner syndrome. And let's talk about some molecular study. The last part of your class. We have some different genetic tests. In biochemical assay, we're looking for abnormal metabolites inside the body. For example, phenylketonuria. For example, we evaluate the salt level in sweat glands in cystic fibrosis. Also, we can detect enzyme deficiency. Okay, we have a lot of examples in our slides. Sometimes we need to evaluate the structure of chromosome. For example, in Down syndrome, we use karyotyping. We can see the big problem, big structural or numbers abnormality in chromosome. But when we want to find small abnormalities, for example, deletion in some areas of the chromosome, we're using fish. What is fish? What you eat? Fish, ماهی. شما چی میگین؟ Simit, not simit, not fish, not ماهی. Simit. In Persian, simit or simit mean some people stick to you and don't leave you alone. Simit, we said. طرف سمجه. شنیدی؟ Semij in Persian, we mean a person come and don't leave you. No, not kind. It makes you nervous, yes. Don't leave you alone. Semij. آره. Sandwich. So sandwich, not fish meat, doesn't mean sandwich or mahi in Persian. Farsi, fish mahi. Fluorescence in situ, fluorescence in situ, hybridization. In this type, we can find abnormalities more than 100 kilobase in size. In fluorescence in situ hybridization, we have probe, known probe. You can buy them. They make in laboratory. You find a probe, you have a probe. These probes are attached to a dye, So when they mix with the cells, the patient cells, these probes go and try to find the region to attach. And when they attach, they become fluorescent. And you can see this light by light microscopy. Okay? For example, in breast cancer, we use fish fluorescence in situ hybridization to find the mutation in CRB4, B2 for HER2/neu. HER2/neu is a gene that will cause breast cancers. In fish, we have known probes that go and attach to HER2/neu, and when they attach, they have fluorescent. They become fluorescent. Did you got it? Did you understand? Yes? Thank you. But sometimes we're looking for abnormal sequence in DNA. PCR, polymerase chain reaction, Sanger DNA sequencing, and next generation sequencing. In some disorders, we have multiple genes which can cause one disease. For example, in Marfan, we have so many different mutations that all of them can relate to Marfan syndrome. In Sanger, in each test, we can just find one gene which is abnormal. But sometimes we need to repeat the test to find the abnormal and mutated gene, which is related in this person to Marfan syndrome. Okay? In next generation sequencing, we can find all the genes, we can evaluate all the genes that related to Marfan. So easily we can find the abnormal mutated gene. Okay? So this one is very expensive and time I'm wasting the time. But this one, next generation sequencing, are lesser expensive and not time what? Yeah, not time costing. Yeah, thank you. And the last one, not these are not for your class. PCR, polymerase chain reaction. In coronavirus, we use PCR to find the infected patient. Do you remember? We use PCR, polymerase chain reaction. In polymerase chain reaction, we know the target cell, we know the target sequence, and we amplify it. when we amplify it, then we can detect it easily. So
The comparative genetic type
it's not for your class. So for PCR, we need DNA polymerase. We need DNA polymerase. We need primer to say to DNA polymerase, come on here, you should bind to this part of DNA. We need nucleotide to make another strand of DNA. We need buffer solution for to have which is enriched to have to optimize the activity in this tube, and we need thermal cycler. Look, this is DNA polymerase. Primer said, come in here and stick here. Nucleotide, come, and we will have another chain of DNA. We amplify the target sequence, and then detect it by denaturation, annealing, and extension. In denaturation, two strands of DNA separate, primer and polymerase come and attach to. So we will have so many, we will have at the end of PCR, we will have so many target DNA that we can detect it. Do you understand? Can I explain it for you or not? Was it clear? Yes or no? Yes? تارا ببین بچه ها فهمیدن؟ لا نفهمیدن. میتونی براشون بگی؟ میتونی براشون بگی؟ ببین نگاه کن. ببین توی PCR ما یه قسمت خاصی از DNA رو به عنوان تارگت داریم. مثلا ویروس کرونا ویروس. خب یه قسمتی به عنوان تارگت ما هست. بعد این تارگت باید بیاد از روش هی ساخته بشه. خب ما نیاز به چی داریم؟ یه DNA پلیمراز داریم که بیاد نوکلئوتیدها رو کنار هم قرار بده و اون تارگتمون رو بسازه. نیاز به یه پرایمر داریم که این پرایمر شناسایی میکنه اون منطقه‌ای که باید زیاد بشه. خب بعد نوکلئوتیدها رو داریم که با پلیمراز میان قرار میگیرن، یه زنجیره دیگه ساخته میشه. خب DNA پلیمراز پرایمر بهش کمک میکنه، علامت میده که DNA پلیمراز بیا توی این قسمت زنجیره بشین و این DNA ویروس اینجا هست، اینو بساز. خب شروع میکنه با ساختنش با نوکلئوتیدها. همه این اتفاقات توی لوله های آزمایشگاه اتفاق میفته که داخلش بافر خاصی وجود داره که این بافر مواد شیمیایی مورد نیاز رو فراهم میکنه برای PCR و یه ترمال سایکلینگ داریم. توی ترمال سایکلینگ ببین اینجا رو، این پلیمراز با پرایمر میاد بهش وصل میشه، زنجیره DNA ساخته میشه توسط چند مرحله. اول از همه دناتوره میشه. یعنی چی؟ توی دناتوره زنجیره از هم باز میشه. برای دناتوریشن لازم هستش که درجه افزایش پیدا کنه. تو اون ترمال سایکل درجه میره بالا. این دناتوریشنه. بعد انالینگه. توی انالینگ
پس اینو ما دی ان ای پلیمریز داریم میاد اینجا میشینه
دناتوره میشه. دی ان ای پرایمر بهش میگه بیا اینجا بشین، میاد میشینه. با نوکلئوتیدها حالا انلینگ اتفاق میفته، یعنی شروع میکنه یه زنجیره رو به ساختن. خب زنجیره ساخته میشه. توی این مرحله باز هوا گرما کاهش پیدا میکنه، سرد میشه. اینجا دناتوریشن گرمه. The end of your class. Yes, you have too much time for rest. Huh? You wanna question? خب تارا این چجوری تموم میشه؟ خواهی بری بیرون؟ You wanna go around? Huh? A 22-year-old man has a sudden loss of vision in the right eye. On physical examination, there is subluxation of the crystalline lens of the right eye. He has mid-systolic quick echocardiography show floppy mitral valve, mean mitral valve prolapse. The patient's brother and his cousin are similarly affected. Which is the genetic defect? What do you think? Mean fibrillin 1. So, Marfan syndrome, subluxation of the lens. Yes, thank you. Marfan syndrome. A 22-year-old premi-gravid notes absent fetal movement for 2 days. The fetus is delivered silver at 19 weeks gestation. The macerated fetus show marked hydrops fetalis and large posterior cystic hygroma of the neck. At autopsy, internal anomalies are seen, including coarctation of aorta, horseshoe kidney, B, D, A, turn, A, E, E, 45XO. What's the disease? Turner syndrome. E. Ladies. Again, a 22-year-old woman delivers healthy female infant after an uncomplicated pregnancy. At 4 years of age, the girl is brought to the physician because of her parents have observed progressive severe neurologic deterioration. Physical examination shows marked organomegaly. A bone marrow biopsy specimen shows numerous phony vacuolated macrophages. Analysis of which of the following factors is most likely to aid in the diagnosis of this condition? Number of LDL receptor, level of sphingomyelinase, rate of synthesis of collagen. B, B, B. What's the disease? Niemann-Pick. You can see it is foamy vacuolated cells, so is lysosomal storage disease. So the answer is level of sphingomyelinase. Number 4. Ladies, answer me. Mental retardation has affected several generations of family, and most of the affected individuals have been males. The severity of mental retardation has increased with each passing generation. Genetic testing is performed, and about 20% of the males who have genetic abnormality are unaffected. Which of the following mechanism is most likely to produce this genetic condition? A. Ladies. Batul, what's your answer? Ladies? A? A, fragile X, 3 nucleotide repeat mutation. The answer is A, yes. Fragile X syndrome. Question number 5. What are these cells? Look at them. Yes, these are histiocyte macrophage. But what is the appearance inside the cytoplasm? Yes, lipid. What is the pattern? Wrinkle? Tissue? No. Onion you couldn't see by light microscopy. It's a fissure that you can see by in microscopy, electronic microscopy. Tissue, wrinkle, tissue pattern. Wrinkle, tissue pattern. So, a bone marrow biopsy is performed and high magnification of the sample shows the finding in the figure. An inherited deficiency of which of the following enzyme is most likely to produce these findings? What's the answer? A. What is the disease? Gaucher. Yes? Thank you. برم تو هوم میخوام برم تو اسلاید قبلی بذارم این تو back of the class. We don't have any back of the class. خب No.
So the questions of the exam, they were same as these questions?
Most of them. Tara. Do you want to watch cartoon? No? Sorry, but you have to watch some cartoon here. صدا داره این؟ تارا does it has a voice sound?
Ever since I was a kid, I've been curious about mutations. I think certain movies may have had an impact. I used to imagine mutations being but there was also a lot I didn't understand about mutations. Let me explain. Many people understand that a mutation is a change of genetic material. More specifically, a change within a nucleic acid. RNA and DNA are both types of nucleic. Therefore, anything with RNA or DNA can have a mutation. That means animals, and that definitely includes humans, also plants, fungi, protists, bacteria, archaea, they can all have mutations. Additionally, so can viruses. Many mutations can be neutral in effect. You can see this codon codes for the amino acid leucine. But if it experiences this particular silent mutation, even though a base has been altered, it still codes for leucine. It did not change the amino acid. Mutations can also be harmful or helpful, but it's important to understand that mutations are random. The organism can't will itself to get a certain mutation. You can learn more about this in our natural selection video, where we talk about how a bacterium may already possess a helpful mutation that allows it to survive an antibiotic. But the bacterium didn't will itself to mutate to develop this certain mutation. Okay, so again, mutations are random, but there can be factors that make mutations more likely to occur. External factors like certain types of chemicals or excessive radiation, or internal factors like an event that causes a problem during DNA replication in interphase. During interphase, cells can replicate DNA before they divide. So let's discuss some different types of mutations. We'll start with gene mutations. DNA makes up genes, and genes can code for proteins that influence different traits. So when a mutation in DNA happens, which specifically means a change in one or more DNA bases, then different proteins can be produced, which can affect an organism's traits. In this example, we have fruit fly DNA. Mutations could include substitution, which means the wrong base is matched. Insertion, which means an extra base, or bases, are added in. There is also deletion, which means a base is removed. Insertions and deletions have the potential to be especially dangerous. Why? Remember in protein synthesis how we talked about how bases are read in threes? Well, if you add a base or remove a base, suddenly the number of bases total has changed. And if you read the bases in threes, depending on where it happened, everything that is read afterwards could be affected. We call this a frame shift mutation. If you look at this frame shift example here, where a single base has been inserted, you can see how the following codons, which have 3 bases each, are now all affected as the reading frame has been shifted. This example shows how it can lead to many amino acid changes. There are also chromosomal mutations. Remember that chromosomes are made up of DNA and protein, highly organized, and they have lots of genes on them. The human chromosome number is 46, with 23 from an egg cell and 23 from a sperm cell. This type of fruit fly here, however, has 8 chromosomes, so 4 came from an egg cell and 4 came from a sperm cell. Some examples of chromosomal mutations include duplication, where extra copies of genes are generated, deletion, where some of the genetic material breaks off, inversion, when a broken chromosome segment gets inversed, which means reversed, and put back on the chromosome, or translocation, when a fragment from one chromosome breaks off and attaches to another chromosome. There's more mutations than what we covered, of course, but the idea is that there are many different kinds of changes that can happen. If a mutation is going to happen, we already mentioned that there are especially vulnerable times, such as during DNA replication. But also there are other times too, like during meiosis. In fruit flies and other animals, meiosis makes sperm and egg cells that can have half the number of chromosomes as the organism. However, sometimes those chromosomes don't separate completely. We call this non-disjunction. This can result in an egg or sperm cell that has too many or too few chromosomes. It's possible for a mutation to be passed down to offspring. Consider a protist with a mutation. Many protists reproduce asexually, and when they divide, the daughter cell can inherit the same mutation. A fruit fly, which reproduces sexually, can pass a mutation to its offspring if that mutation is found in the genetic material of the sperm or egg cell. You may wonder why we've been mentioning fruit flies so much. Well, they're awesome, but also it turns out that fruit fly mutations and how they are inherited are frequently studied. We have some links in the video description for more info. These studied mutations can also occur in humans, and we'll give a gene mutation example of a substitution in the case of sickle cell anemia. First, a little background. Hemoglobin is a protein in your red blood cells that helps you carry oxygen. But in the disorder sickle cell anemia, the gene that codes for hemoglobin is mutated. If you inherit 2 copies of this gene, one from each parent, you can have this disorder. This disorder can make it difficult for your red blood cells to carry oxygen because the shape of the red blood cell is affected from this mutated hemoglobin protein. This can lead to anemia and other problems. Well, unfortunately, there is not a cure for this disorder yet. The good news is that treatment for this disorder has greatly improved. Another thing to mention, if an individual only inherits 1 copy of the mutated gene from one parent, they are a carrier, but they don't officially have the disease. Usually they do not have symptoms, but those that are carriers appear to have a protective factor against malaria. Malaria is a disease caused by a protist that can be transmitted by mosquitoes. These individuals can still get malaria, but their symptoms are often less severe. We should mention that studying mutations and genetic disorders is a large and important field right now. Genetic counselors work to help families that may be affected by genetic disorders. If you have an interest in learning more about careers related to this topic, please check out some of our further reading suggestions in the description below. Well, that's it for the Neva Sisters. I'll remind you to stay curious.